I'm here to share two things. One, through my work, how neurons take stress and actually show resilience. And two, through my work and how I worked at IISER Tirupati, how we built up certain things from nothing. Because IISER Tirupati started in 2015, I'm one of the first biology faculty who entered a room with really nothing and brought a lot of things today, stands for everything that I'm going to show you. When I just walked this morning besides the beach, I could smell the Shenbagapoo after several years. All the Malipu and Shenbagapoo. Shenbagapoo is this flower very native to this region. And I've smelt this strongly during my childhood and maybe a little bit when I was in Bangalore in the Champige Road. After that, during my Germany life and where I am now, I didn't smell it. So this triggered me again to my childhood and made me feel alive, thanks to my olfactory neurons that I actually felt that, and to my cortical and hippocampal neurons that engraved this memory deep inside. Then I went back to the guest house, had the favorite dosa. The taste stuck until even before lunch in my taste bud, thanks to 8,000 and odd taste buds that this is made incredibly possible. Do you know how long these taste buds live? From 2 weeks, maximum 3 weeks. So from 2 days to 3 weeks. When a child is born, it has about 10,000 and odd taste buds. As we age, it reduces to maybe 5,000. So probably I'm standing with 8,000 and odd taste buds. Still, I could carry the taste for so long. Again, the morning, a vast blue ocean never stops amazing me. That blue sky and the ocean, which gave me a great feeling from my visual perception, and the smell of salt, all put together made me feel alive today again. It was possible because I could see through my retinal neurons. You see an image there. I smell through my olfactory neurons, and I hear and listen through my auditory neurons. So a human lives through the perception of touch, smell, seeing, hearing, and tasting. This perception is made possible only by small warriors called neurons who take back all the messages from the environment to the brain and create a long-lasting memory in the hard disk called the brain. It can be short-term or long-lasting, depending on how we train the brain, and that's what we're going to talk about. As I just said, there are several kinds of neurons that you can see, and what you can also appreciate is they are all very individual. They all look different in the shape, and they all are doing different functions. Although they are all neurons, they carry their individuality, and each of them do very specific functions, and more interestingly, the neurons are non-dividing. When a child is born, it has a huge toolkit, several million neurons with 500,000 million synapses. What are these synapses? These are connections that connect two neurons, which basically transmit signal from one to another, enabling the communication through the form of electrical impulses. Speaking of which, the brain functions in an electrical impulse that is enough to light a bulb. Maybe not so bright, but bulb enough for basic functioning. Interestingly, as soon as the child is born, the brain starts thinking, do I need to keep so many neurons or do I need to cut down? One fundamental secret about the brain is that brain and the immune system are the two organs that continue to develop even after birth. Brain actually is the first organ to start developing in utero. In utero, in uterus, in the mother's womb, that's your first home. So it starts developing first, but keeps on developing until long time. It's not like heart, it's not like lung, it's not like any of them that actually has a rhythm as soon as you're born. Whereas brain continues to interact with the external world and brings all good, bad, and ugly to the inside, and as I said, keeps creating short-term and long-term memories. Now, as as I just mentioned, if you do not keep training the brain, you're going to basically lose out on neurons. So it is in the hands of the child, it is in the hands of the environment to keep the child challenging, both the environment as in the parental environment and the child himself or herself, to keep challenging with new challenges and to train and to let the brain grow stronger connections. So to do that, new tasks are important, which is called as the cognitive period. During this period, if there is not enough challenges, the brain doesn't want to waste its energy and it thinks this is an unusable toolkit and I'm going to remove the excess neurons in a process called synaptic pruning. Most of you know what is pruning. You go out and there's a lot of grass, you try to remove and you prune the grass. It is the same phenomenon. The brain doesn't want to waste energy. So it says, okay, I'm not going to waste myself in keeping all these connections. It's not being used for a long time. I'm going to prune it. Therefore, the first 20 years of life is very incredible, very important. So you continuously learn new things, novel, challenging things. Subject yourself to very important challenges. You do not limit yourself, you push your limits. Learning doesn't mean you learn new things every day, but a consistent learning. So you learn, you repeat, you learn, you repeat, and that's how you actually train your brain to connect. Because the first time you expose the neurons to something, it is perceiving and it is keeping the information. It is processing whether it has to keep it for long or not. The second time you do the same, the brain thinks, aha, it makes sense probably, I'm supposed to keep it. The more times you keep doing that repetitively, and that's when this is going to be formed in the memory hard disk. Otherwise, it is in your working memory or the short-term memory. If you want to build it in the long-term memory and in your hard disk, you need to repeat. You need to slowly learn, you need to focus learn, and you need to consciously learn. The eye is subjected to very many insults from the external environment. It can be the light intensity, it can be the pressure around, it can be the temperature, it can be anything around the environment. Whereas the brain is snuggled inside the cranium, kept very beautifully inside, protected like a chest, you know, it's a chest and then it's protected inside because of two reasons. It needs that protection on one hand. Why it needs? Probably because it is very vulnerable on the other hand. From the scientific, from the biologic point of view, see, it may be vulnerable. It's not as tough, it's not as resilient, it doesn't cope up to the stress as much as maybe the olfactory neurons or the retinal neurons or the dermal cells for that matter. Olfactory neurons are exceptional in terms of its regeneratability. Same is true with retinal cells. To a large extent, you can talk about taste buds, but not gustatory neurons. That brings us to the question, do neurons regenerate? Not all. With that question in mind, I started establishing my work lab at IISER Tirupati after a break of 3 years for my maternity, and then wondering what question to ask which would be relevant to my country. I was trained as a cerebellum scientist. Ataxia was my disease model. But in India, we do have ataxic patients, but not as much as we do have diabetic patients. Not as much as we do have diabetic-related vision impairment. So when I started off, I asked my question, what should I do that is impactful for my society, for my nation, and my outcome actually brings something really important. And that's one side. The second side is we went on to work with Veterinary Institute because I just told you that we started off in a place where it was really nothing. So we went to Veterinary Institute to collaborate and we asked them if we can have some brain tissues with which we can explore. So we brought the brain tissues from mammalian adult brain and also the retinal tissues and systematically cultured these, from the hippocampus, from the cortex. So these are different regions of the brain. So the cortex is here, the hippocampus is more deep-seated, and the cerebellum is behind, spinal cord. So different regions of the brain and also the retina. We cultured them in vitro. Now the global research already tells us two lessons. Many scientists have tried doing this and they have clearly shown that the outcome is dicey. Not every neuron is culturable, but we still said we want to look at it in our own hands and setup and see what is this. And to our beautiful surprise, we could actually repeat what most people have said, which means we are doing well in the way, but to a next extent, we could grow the retinal neurons in culture, in the laboratory setup for about 12 days, which provided us a very interesting platform to ask very many questions and see questions like what will happen to these neurons if I subject them in different glucose conditions. I'll come why I'm doing that. And if I subject them to different oxygen conditions. These are the two metabolic stressors in the body. I just talked about the environmental stressors, but internally, these are the metabolic stressors. Every time throughout the day, all of us go through variations in glucose. I'm sure 80% of the population here, or 60%, because I do see a lot of young folks as well, at least once did your blood test, and now we know what it is. So when you do blood test before, after food, you know the variations. So this already tells that our body continuously goes through glucose variations, which puts some of these sensitive organs to the glucose tremendously. Now, under this condition, what happens to the neurons in the long term? Nobody really knows about this. So we started asking these questions. What we interestingly noticed was, if you subject these neurons to different stressors, glucose or oxygen or any protein that is important removed. So in biology, this is the way often we do experiments. If I want to know the function of, let's say, this TED platform, I remove the main organizer and see how it functions, right? Then I remove 3 organizers. Then I see, is the platform still fine? This is exactly how we function. So here as well, when I want to know what certain proteins do to the cells, we do what is called as knockout strategies, we remove them. So this you can really see that one image on the side looks more roundish with some processes. On the other side, you see neurons with elongated process with shapes that are changed and so on. So this is what we observed when we subject cells in different glucose conditions and or removed an important protein. An important protein that we have worked in my lab is called contactin-associated protein, and we were very excited to take it further. Although this is a huge protein, which means typically of no commercial value, because if you want to make any protein as a target for medicine, for drug, they have to be typically small, because then it is handleable in size and they can cross what is called as the blood-brain barrier. This is one of the challenging parts for any neurologist, for any drug discovery people, to let any molecule cross the blood-brain barrier. This is a sanctity area that everything gets filtered in the blood-brain barrier, and only very small particles like the glucose, like the solutes, can go to the brain. So we know that it's a large protein. It probably will not be a great target, but still understanding the phenomenon, understanding the factors that are stressing the neurons in different conditions are very important to understand how neurodegeneration can be delayed or neural regeneration can be promoted. The same protein, to our surprise, when we collaborated with NBRC, National Brain Research Center, and when we collaborated with LV Prasad Eye Institute in Hyderabad, we took the sections of mouse retina from NBRC, diabetic mouse retina. There also, we saw this protein was very reduced significantly, stating that in the diabetic eye, this is going down. Is this a marker? Can we use this protein or proteins like this as markers to detect diabetic retinopathy faster? That's a question that we are addressing currently. Similarly, also in the human diabetic retina, this protein goes down. So what you see here on the left side, this is all the control and normal patient, whereas there it is a diabetic patient. Clearly, 2 things emerged from our studies, that neurons respond to metabolic stressor in their own way, which we should understand better. And what we now know from other studies as well is that between the conversion of healthy retina to a diabetic retina, there are several processes. There are several stages that lead to the conversion from healthy to diabetic retina and then the blindness. Intervene at the right time, this can be regenerated. What is also widely accepted that the retinal neuronal layer thins down. So this is a widely accepted concept by the ophthalmologists and clinicians, and that indicates already a neuronal layer degeneration in the eye, which can be a marker towards diabetic retinopathy. So we are addressing how we can reverse this phenomenon, by which pathway, by manipulating what protein we will be able to reverse this. In India, this can be a very alarming, slide for us here, because you see that the highest number of diabetic patients are in the southernmost part. In fact, in order not to threaten the population, I did not bring another slide which tells you how in the last 30 years we have transitioned from a normal population to heavily diabetic population. This clearly tells us we have to step back and think what we are doing. I'll arrive at that in a couple of minutes. But in India, the number of diabetic patients are clearly increasing. Earlier, it was the disease of the... Now, the mid-40s, early 40s are also in the burden of pre-diabetes or diabetes. That means somebody with an onset of diabetes at 40, 45, who will live 80, 85, has higher of neurodegeneration risk. Unless we think and take steps, we might land up in having a population which lives longer but not functional brain, which is what we don't want. We have to be healthy, not just physically moving or doing things, but mentally comprehending our society well and being able to interact. So that's why we try to address this important point. So this also shows the conversion rate in the next 5, 10, 20 years, as you see that it's increasing. The neurons do take stress individually much longer than all of us can take the stress as a human self. They are much stronger than us for sure, but they also have break point. So socializing, learning new activities systematically, and the most important, you do not subject your neurons to all these glucose insults that they don't like. You help neurons to be more resilient, and you yourself turn to be resilient. Thank you.